And we've also discussed it earlier, as long as the recording works. If you want to go back, you can go back and look at that. But this is where they said, okay, what can we say interpretively about this? Well, we can rank accessibility of each patch on the protein. We can rank polarity. And we can see where this particular patch is so important. How does it rank among all the patches on the protein? It's highly accessible, but it's lowly polar. And this is where we got the conclusion at the end. Also, notice what's not there. When you're reading a paper, sometimes you'll notice that what's not there is only 19% of the surface is capable of hydrogen bonding, compared to 37% on average. The way you have an accessible but non-polar surface is you lose the ability to do hydrogen bonding relative to the rest of the surface. So the one thing not to say about this is that it works through hydrogen bonds. Yeah, it can, but it's not the major way that it works any more than it's the major way that it works through salt bridges. So the rotation, like the northwest corner rotation that we saw, that did form some hydrogen bonds, probably, if we can trust the resolution. But there were other things that didn't depend on it. So what they have is it has a bunch of like 8 or 9 diverse residues that has a groove in the middle with methionines in the bottom that can bind a lot of different things because sulfur is good at that. And then it has a bunch of different residues around the edge. That's why I think it's so cool about this. You actually have salt bridges in one, you have hydrogen bonds in another, you have just hydrophobic things rotating on the northeast corner. I think we saw that. And it adds up to be something that is just very bindable. It is a conformational change, but it's a rotation. It's not the backbone changing. The backbone's staying the same. So it's a small conformational change, but a lot of small things can add up to make a really good binding site. So the only thing I can say is that you can look at, they colored where the highest average accessibility and lowest average polarity was. And really you get this deep groove that's colored red, and nothing else has a deep groove that's colored red. That suggests that this is important. You can also look at other red parts. And what I find interesting is the red part here, guess what? That is the FC receptor binding site. So it looks like that the FC receptor might even follow the same rules. It's just it can bend the rules and it has to bind at a different point. So the methionine is really important and a cool residue. It's unusual to see both of those. And what the answer is, and I'm sure this was added in review, when you take stuff to reviewers, they love to ask you to do another experiment. In fact, Tracy Delgado just submitted a paper, and they always come back and say, oh, can you do these 3 or 4 more experiments? Which is fine usually, but they assume that you have like a whole lab up and running all the time. When you're working with undergraduates, you don't necessarily have the ability to do this. But they were a whole lab, so they were at UCSF, they were able to do this. And they were able to actually do mutations and see when we mutated these important residues that the binding went away. And that's a good control. It looks like this came from reviewers. Again, probably doesn't fit into the short talk. And so this leads to the over thing that we're walking a fine line. We're not depending on any one kind of interaction. We have flexibility. The flexibility is side chains rotating, not backbone moving. So you can ask questions like, could this be used as therapy against rheumatoid factor? Well, maybe. It's small, but peptides are terrible at being therapeutic agents. There's lots of proteases in the body. You could have immune reactions, things like that. But it's theoretically possible. If you want to propose something like this, you can. You feel free. Believe me, I'm not going to. I will love the creativity that you can show if you can have some kind of thing. This is just me wondering, but it probably has a problem with it. Another problem is because of the FC receptor, no nursing mothers should probably apply. It might get into blocking the baby from getting antibodies. So the very last thing I want to say is, Savannah, you have a special in on this for your paper, because your paper finds the FC domain. They created a biosensor that can detect 3 or 4 different proteins. One of the proteins was the FC domain. So you should check that out and you should see, does it bind at the same site? I think they probably use protein G or protein A. I literally worked in the Baker lab and I actually studied protein G when I was doing the experiments there. But that's why I'm almost sure that it's probably protein G, but at least this has a little bit of a connection for you. So yeah, look back at this when you're putting together your protein design paper. Always focus on what the question was, always focus on the structure, and always focus on how they know it worked. Like, show us that. And I can also put this online if you want to see it, but it's a good thing to take a picture of. I do agree with that. Okay. Yeah, and here are my answers to what those 3 questions are. James A. Wells is now a big name, and we're going to see what else he has done. He's back at UCSF now. I don't think he's with that company anymore because the company has come and go, but we'll talk about his research as we go through. Just your due date for next Wednesday. This is posted on Canvas. I have 2 videos for you to watch. It's about small molecule design, but they actually has to do with protein design as well, and it helps us to make a bridge. We'll talk about what James Wells did after this. And so the links, I don't think you want to go to the links, just go through Canvas and look for the links there. I tested them, they work. In a couple weeks, I'll remind you of this. We'll be going through another paper. This is a James Wells paper that's very different. And we'll be doing the same sort of thing. So I'll be giving you the paper. This is just a much different paper, but I will put it on Canvas. The main thing is to remember we don't meet next Wednesday, but we will meet, and we don't meet Friday because of Good Friday. It's just that this is where the James L. Wells talk. So we will next meet in, is it a week and a half on Wednesday, and we will do our presentation. So do let me know if you have questions about the presentations. But mostly, yeah, that brings us to this, to the end of